Hello and welcome to the lecture 1 of our bioinformatics series. Today we will take you on a journey from the raw sample of our bacterial isolations all the way to the final report using the whole genome sequence. I'm your instructor Linda Khalid and I'm a bioinformatics researcher and a microbiologist as vast experience researching sequence all the different genomic biological insights. So by the end of this video, you will understand what basically the whole genome sequence is and what is antimicrobial resistance and what are the how whole genome sequence is used in AMR research, how we can decode the drug resistance bacteria to produce publishable results out of this sequence. Uh, these 5 goals will guide our entire session today. What is whole genome sequence? Whole genome sequencing is the process of reading the complete DNA of an organism in a single experiment, first used in 1995. It gave us 100% genome coverage across more than 4 major sequencing platforms. History and the sequencing methods. Whole genome sequencing was pioneered by the J. Craig Venter and Ed Smith in 1995 when they first sequenced the Haemophilus influenza and M. genitalium. Today, the most widely used method include the Illumina for short reads and Nanopore for the longer reads, and the shotgun for the random fragmentation and reassembly. How whole genome sequence works. The whole genome sequence process began by fragmenting DNA, which we isolated in our wet lab experimentation, the sequencing those fragments into reads, and then using the bioinformatics tool to detect the overlaps between them. Finally, those reads are assembled together computationally to reconstruct the complete original genome out of. So here are some of the common sequencing platforms. The 3 major platforms are Illumina for high sequencing accuracy and shorter reads, and Nanopore for the longer read, best suit for the plasmid and the structural analysis, and the Pace Bio for producing the complete genome assembly. Each platform has its own strength depending on your research goal. Type of sequencing data. The whole genome sequencing generates 3 main data types. First one is the FASTQ file containing raw reads in the quality score. Second one is the FASTA file, the assembled genome with contigs in. And the third one is the annotation file that identifies the genes, rRNAs, tRNAs, other functional elements. Together, these form the complete picture of a genome. We'll see now the whole genome sequence analysis of the multi-drug resistance and extensive drug resistance pathogens. Using the whole genome sequence, we can identify antibiotic resistance genes and detect the virulence factor. We can find the resistance carrying plasmid and perform the MLST typing for the different strains classification. This comprehensive analysis is especially powerful when dealing with the multi-drug resistance and extensively drug resistance pathogens. Now we will see why conventional methods falls down. Conventional methods only target limited genomic region and often requires multiple and repetitive tests while making outbreak very difficult. While the whole genome sequence solve this by recovering the entire genome in a single assay with a single nucleotide resolution for size trans. Questions that a whole genome sequence analysis can answer. The whole genome sequence can answer the most critical questions in the field of microbiology, in the field of medicine, like which resistance genes are present, which virulence factor cause the disease, and how pathogens are spreading and evolving globally, transformed complex genomic data into actionable biological insights. Here are some of the major applications of the whole genome sequence. Whole genome sequence is applied across four key areas. First one is the clinical diagnosis for the resistance and virulence profiling, public health for AMR surveillance, research for the comparative and evolutionary genomics, and the epidemiology for the outbreak investigation and transmission tracking. This was all about the whole genome sequence. Let's wrap up our lecture number one. Thank you so much for watching. If you find this video helpful, please like, subscribe, and share so more people can benefit from the content. Thank you.